Hi, my name is Jane Picarelli, and this presentation is going to be about the major and the minor fluid compartments of the body, as well as their ionic composition. On the upcoming slides, I will be describing the different fluid compartments in detail, but to start off with, I just want to show you this graphic organizer that I created, which will be referenced throughout the presentation. So the two major fluid compartments in the body are the ICF, which stands for intracellular fluid, and the ECF, which stands for extracellular fluid. Now, the intracellular fluid, which is the first main compartment, consists of the fluid that is inside the cells. So intra means inside, and this fluid is inside the bodily cells. And in total, it actually accounts for 2/3 of the body's total fluid composition. Although this seems like a lot, when you take into account how many cells there are in the human body, this becomes quite reasonable. Now, the extracellular fluid, that's going to be the other main compartment, and that consists of all the body fluid that is not contained within the cell. Now, there's 4 types of fluid that make up the minor compartments of the ECF, and those are going to be the plasma, the interstitial fluid, lymph fluid, and transcellular fluid. We will dive further onto this in the next slide. So to the far right of this image, this green portion, this all represents the intracellular fluid, so the fluid that's inside the cell. It is very different in ionic composition from the extracellular fluid, all that fluid that is not within the cell. And that's going to be because of reasons that we'll soon be discussing. It is important to know for now, though, that the extracellular fluid, again, all of this, is separated from the intracellular fluid by the cellular membrane, because recall that ICF, intracellular fluid, is the fluid within the cell, and ECF is the fluid outside of the cell. Therefore, there's always going to be that membrane that divides the two compartments. So now the pink, the blue, and the yellow portion of this image all compose the extracellular fluid, so all of that. To the far left is a representation of the blood plasma. Now, the plasma is mostly fluid, and it's found in the vessels and is part of the body's circulation. Because plasma is in blood vessels, including the very tiny and permeable capillaries, the interstitial fluid, which is here, and the blood plasma are easily exchanged with one another. They're very easy to go through those capillaries because the barrier to those is the capillaries. Now, interstitial fluid, which is represented here in blue, is fluid that is not inside of the cell, but still is able to make some exchanges with the cell. Lymph fluid, which is depicted, it's the fluid going this way, and here I titled it lymph, but it's literally going from here to here. That lymph fluid is the interstitial fluid that is being sent back to the plasma after it's filtered by the lymphatic system. So now, transcellular fluid, which is represented over here, this is fluid that was originally secreted by specific cells, but now it encompasses a particular body cavity where it serves a specialized role. And that role, depending on where the cavity is located, does different things. For example, you have heard of cerebral spinal fluid. That surrounds the spine and the brain to protect it by acting like a shock absorber and a lubricant. That's a transcellular fluid. Other transcellular fluids include synovial fluid, pericardial fluid, intrapleural fluid, and intraocular fluid. Now, in the previous slide, I briefly mentioned the barriers between different compartments. For example, I said that the barrier between the intracellular fluid and the extracellular fluid is the membrane of the cell. Well, it turns out that the type of barrier between the compartments has a dramatic effect on the consistency of the types of fluid, including their ionic composition. If you look at the image on this slide, you will see a capillary, all of this. You will see an interstitial compartment, and you will see a cell. Keeping in mind that all fluid that is not inside of the cell is still considered extracellular fluid, you can see that separating the intracellular fluid from both types of extracellular fluid, meaning the plasma and the interstitial fluid, is the cell's membrane. So because this barrier, the membrane, is very selective in what materials it allows to be exchanged with extracellular fluid, the consistency of the ICF and the ECF is very different from one another, and we will see just how different it is on the next slide. Now, since the minor compartments of the extracellular fluid, which in this image is the interstitial fluid and the plasma, are not separated by cellular membrane, but instead are only separated by a highly permeable capillary, there is a lot of exchange between the two compartments, and therefore the composition of the interstitial fluid and the plasma is quite similar. Both of them are very rich in sodium and chloride. The main difference in their constituents is that plasma contains plasma proteins, which cannot be exchanged with the interstitial fluid because they are confined to the plasma and cannot pass through the capillary, and therefore interstitial fluid lacks these proteins. Other than that, though, the plasma and the interstitial fluid are extremely similar. I created this image specifically to detail the differences in ionic composition between the extracellular fluid and the intracellular fluid and highlight how those differences are made possible. So the first thing that must be noticed is that the extracellular fluid is rich in sodium ions, which are shown in green, and chloride ions, which are shown in yellow. It also contains a minimal percent of bicarbonate, but that's not really significant enough to represent in this particular illustration. So now remember how I said that the ECF and the ICF were very different in ionic composition? Well, if you look at the intracellular fluid, so the fluid that's inside of the cell, you will notice that it consists mostly of potassium ions, which are shown here as gold triangles, and phosphate, which is shown as purple circles. And there's some proteins also in there that have a negative charge, and those are shown as large brown circles. Now, the major differences in the composition of these two compartments is due to two main factors. So the first main factor is that not all substances can readily pass through the membrane. And therefore, we talked about how the membrane is the barrier between the ECF and the ICF. So because not all substances can readily pass through the membrane, this barrier maintains the distinction between the two types of fluid. So the second factor is that the cell contains a type of pump called the sodium-potassium ATP-base pump, which I have represented in this image as a pentagon. So here's that pump. Now, this pump exchanges two potassium ions, so there's two potassium ions, for three sodium ions. So this pump allows for the potassium to travel inward and go into the intracellular fluid, and this is why the ICF has such a high potassium concentration. And now the sodium, shown here, is going to travel outward and into the extracellular fluid. And sodium tends to attract chloride as its complementary anion because sodium has a positive charge and it has a high affinity for chloride. So that is why the ECF, the extracellular fluid, all this over here, winds up consisting largely of sodium and chloride. So these two factors being the membrane and that sodium potassium ATPase pump, those maintain the differences between those major fluid compartments. Now, as I stated, the cell has negatively charged proteins too in the ICF, and these help attract the positively charged cations. So it's important to note that differences across the membrane, differences in charges across the membrane are what leads to cell excitability. And that's in the form of action potential generation, which is how your cells communicate. So therefore, it's very important that the ionic composition of the different types of fluid and their barriers are maintained. Due to the fact that the interstitial fluid of the ECF and the plasma of the ECF can freely exchange any material except for plasma proteins, as we recently talked about. The composition of the plasma and the interstitial fluid is very similar and can influence the other. So for this reason, a change in volume of the ECF can influence blood pressure. So most central to this concept is salt balance, and because water follows sodium in a sense. So the results from the following peer-reviewed study provides a practical application of just how important maintaining proper fluid balance is for human health. And the scholarly article that was published in the New England Journal of Medicine performed a study known as the DASH study, and it was done in April of 1997. So the aim of this study was to determine if nutrients, including salt, play a role in blood pressure pathology in order to eventually come up with an optimal diet to prevent hypertension. So the study itself was performed over a 3-month basis, and there was 459 adult participants. The adults who participated had to start with a systolic blood pressure that was less than 160 and a diastolic blood pressure that lied somewhere between the 80 and 100 range. So the first 3 weeks of the study, the participants were administered a diet that was designed to mimic the average American diet, and this was the control diet. For the remaining 8 weeks of the study, at random, individuals were assigned to either continue on the control diet, eat a diet rich in veggies and fruit, or have a mixed diet which has a high fruit and vegetable content but has reduced fat and reduced sodium amount. The reason it was important to have a set amount of sodium is because sodium itself can alter blood pressure by quote unquote dragging water molecules with it, therefore increasing volume and increasing blood pressure as the body retains water. So the results of this study found that for those individuals whose baseline had already been elevated, because they did have some participants in another group that they started with an elevated blood pressure, that there was 133 of them, that the combination diet reduced their systolic blood pressure by 11.4 millimeters of mercury and their diastolic blood pressure by 5.5 millimeters of mercury. So the rest of the participants we talked about whose baseline blood pressure was already relatively normal showed that their average systolic reduction was 3.5 millimeters of mercury and the diastolic reduction was 2.1. So overall, the combination diet showed the most clinical significance in maintaining healthy blood pressure in adults, and it had maximum effect on participants who were already hypertensive. This makes sense due to the nature of the American diet and how high in sodium it is. I would expect these results, and this is a picture straight from the journal article showing the results as percentages of the 3 different groups of participants, or the 3 different groups of diets used.